Yeah, so Dan tried to convince you that the way forward is large-scale GWAS analysis. And what I want to do is basically talk a little bit about the empirical evidence that we have so far. So, of course, people have tried that, right? So there have been a couple of initiatives that have looked at things like cognitive performance, personality, and even things like self-employment in relatively large samples that are, orders of magnitudes larger of what people have worked with in the candidate gene literature. And, most of these efforts, they came up empty-handed. And of course, this led to a lot of debate in the community, a lot of frustration. So people really started wondering, what is going on? Why don't we find anything? Is it because the effect sizes are so small that with all the data that we have available in the world, there's just no hope of actually identifying something? Is it that the effects are so complex that our simple linear models are not able to pick them up appropriately? Is it that there is massive G by E, which is making it difficult for us to identify main effects? Or is it the case that the twin studies and all the heritability literature is just totally off, right? So people have really wondered about that. And I think the educational attainment project that we did in the SSGAC helped to get a little bit closer to some of the answers to these questions. So first of all, I think in this study what we've shown is that, first of all, GWAS can actually work if you have a sample size that's large enough, even if you look at a biologically very distal and noisy measure of behavior. And also, it's, as far as I know, the first study that gives reasonable estimates of effect sizes of a single SNP on an outcome like that. So it helps us also to calibrate a little bit our beliefs and expectations for future research. So what we did a study on 42 discovery cohorts, and then we had an additional 12 independent replication cohorts. So the discovery sample was around 100,000 individuals and another 25,000, 30,000 for replication. We looked at years of education, and we looked at a binary trait, so whether people had a college degree or not. And then we basically just replicated the standard analysis protocol that has been developed in genetic epidemiology when the medicals are running GWASs on medical traits. And what we found were 3 SNPs that reached genome-wide significance in the discovery stage. And all 3 of them, they actually also replicated. And interestingly, the effect sizes that we found for these SNPs, they were really much smaller than most people expected, especially people in the social science literature. So the largest effect that we found of a single SNP on years of education had an R-square of 0.02%. And the strongest odds ratio we had for college was 0.9. So if you look at the betas and the p-values in the discovery and the replication stage, then you see that they're not drastically different, and clearly the p-value is getting even lower in the combined stage. So if the R-square of a single SNP is so small, that raises questions of what could we possibly learn from that type of research, right? Well, one way of answering that question is to ask a different question, which is how much of the variance in a trait can you explain by looking at a lot of SNPs jointly? And geneticists are doing that with something that they call polygenic scores, which are basically just a linear index where you look at the SNPs that have been measured for an individual, you weight these SNPs with the outcomes of a GWAS, maybe the betas, maybe some other method, and then you just form a linear score. And
why don't you use the individual SNPs in a regression? You have huge samples here.
Right. So, but now we're talking about.
You're aggregating somehow into an index, a scalar index.
Yeah.
Why not just use each individual variable like a multivariate regression?
Well, when we're trying to okay, so the polygenic scores are really they're used for prediction out of sample, right? So they're not discovery. This is prediction out of sample, and basically, yeah. So I think what you're getting at is probably more regressing the SNPs on the phenotype. Yeah. So that is a different problem. Right.
A million SNPs, right?
Yeah.
When you think of aggregating, there are a whole set of techniques that we could use.
Right.
Lower dimensional regression.
Yeah. Yeah.
Using principal components earlier.
Right. Right.
You can imagine that linear or nonlinear versions of that, right?
Yeah.
I'm just
Yeah, yeah. No, yeah, it's totally clear. So I mean, the methods that we're using in genetics here at the moment, they're very, very simple. That's totally clear. So absolutely.
So I think my understanding is that there's basically two constraints to doing more sophisticated kinds of shrinkage or whatever, dimension reduction. One is computational, and the other is that the results, because you can't pool the data, you get these essentially, you get OLS results from the individual level cohorts. So you have like 50 sets of regression coefficients, and I don't know. I don't know of the methods for taking that kind of data and applying these dimension reductions.
So you don't like you don't have the same SNPs on the same individuals. You're saying you have one sample has one, the other has something.
No, they have the same SNPs.
Okay, then why can't I just run this regression with all the
IRB.
Yeah, I understand. So I could then have 50 and I can measure pooling those samples. So it's a more sophisticated question. I run 50 different regressions and then do a weighted regression, which weights all 50.
So that's exactly what that's exactly what the score is. You take the meta. You take each of the 50 sets of coefficients. You meta-analyze the data to get a single set of coefficients for all the SNPs, and then you take a weighted average of the genotypes using those coefficients as the weights. So it's literally constructing, what would be the predictive equation from the regression. That is the polygenic score.
Yeah, I think he's saying instead of going to a scalar, you go to a vector of
individual level.
Of different predictors.
Yeah, I have a vector of however many the principal components I have from the summary measures, and I just have 50 different regressions and pool those coefficients.
Well, you couldn't just use the principal.
In all samples, right?
But the principal components wouldn't be the right thing to use as
No, not principal components. Some data aggregation. It could be lasso, or it could be any one of a bunch of ways to think of low dimensional.
No, but that's captured. All of those are going to reduce dimensionality with regard to the genome,
Right.
but not with regard to the phenotype. I mean, there's a leap of faith there, right?
Well, if you could do lasso,
Well, I thought the phenotype was the same across the study.
it is.
Right.
No, but I mean, imagine you are dimensionally reducing variance in the SNPs
Right.
in a way that produces a bunch of vectors which are worse correlated with the phenotype instead of better.
But I'm predicting it on the basis. I'm choosing a regression.
Yeah, Jim's suggesting
Okay.
doing lasso
More like
regression
lasso.
in
Yeah,
each
okay.
sample or something.
Right.
Okay.
So I do it in each sample.
So the one difficulty with that, honestly, is that the analysts who do these kinds of analyses are not very statistically sophisticated. So they actually, and the software doesn't allow doing things like lasso, and they know how to run the software.
That's your opportunity.
Yeah. So this is actually something that we're working on going forward, but it's not feasible given the kind of available software and.
Okay, just to ask you.
Tomorrow I'll actually discuss lasso in this context.
Yeah.
However, there is a problem that most collaborations cannot pool data in such a way to allow that kind of simultaneous analysis of the whole data set. So the IRBs that individual volunteers sign generally don't allow them to literally pool all their data, which is why these guys had to do
I
it.
mean, you wouldn't pool them. Like you could analyze each of them separately and then pool them that way because the samples are independent, right?
So
I do like a super GLS across however many samples I have.
to use the most effective algorithms, you would like to have access to all of the data at once.
So that I understand, but I'm just saying if you're less stuck with what you have, you can still do a sample because some of these samples are pretty big already by themselves.
Yeah. So one of the things actually we're working on that currently is trying to come up with ways to implement lasso-like output using these uploaded regression coefficient data. But
So, and again, this is not a problem unique to you guys either. I mean, HGO, I know people are trying to fund research and coming up with ways to pool data anonymously, as it were, or in a way that it only makes sense, in the cryptanalytic framework, so you don't ever get the data out again, but you do get the results out. You have to do. It's really weird because in addition to the IRB problems you're noting here, there's other problems too, which is that sometimes if your data comes in contact with someone else's data that's consented differently, so they're consented for farmer use and you're not, we can talk later, Jim. It's a nightmare. Yeah, I don't get those years back.
If I could advertise, one of our colleagues at BGI has rewritten Plink and sped it up by orders of magnitude, and he's also implemented Lasso in Plink. So if you get Plink 2.0, you can run Lasso on your data.
That's great.
Is that downloadable somewhere?
Pardon me? Yeah. It's
Google Plink 1.9 and you just get it.
Is it comparable to canonical correlation?
Canonical correlation.
Not linear. Non-linear. Without the scaling.
Can be non-linear,
Yeah, but I don't think you need the scaling issue. I mean, you have the scaling issue in canonical correlation. Here they have the scale set for the phenotype. Right?
Yeah.
So it's basically liquid regression rather than more like a principal component problem.
No, I would try, if you want to maximize correlation in two sets, well, the other set is one phenotype, right? Or is it the set of measures for the phenotype?
Well, I thought they were running one phenotype at a time.
Yeah. Basically, we're just looking at years of schooling.
Yeah. Right.
Okay, so if you construct the
No, but that
polygenic
raises another
score in the
question,
standard,
which is if you do have this vector of outcomes, why don't you analyze? So if I find that the whole vector, suppose that I had these things that they really were independent, each of these phenotypes, which they wouldn't be. And if I found that each one was statistically insignificant, but 500,000 of them were all positive, then that by itself, there are various combining functions, statistics you could use.
Yeah.
Rodrigo's an
Which
expert.
maximize the correlation with your.
Oh, but the Pesser effect, right? Just these things.
That's why we're hoping Rodrigo's gonna work with
Yeah.
us to make this stuff happen.
No, we've discussed, we've used this in a very different context.
But some of this
Go ahead,
does get
Rodrigo.
computationally really expensive.
Yes.
So maybe, Jim, are you saying take the covariance matrix of these things and optimize your explanation of that with the genes?
Well, no, not so much as that. I'm just making a very simple observation. Suppose that all these phenotypes were independent.
Well, they're not, but okay, yeah.
I know, they're not. So this is more complicated. But the point is, you can imagine each phenotype as being like an independent Bernoulli tribe under my scenario.
I see what you're saying.
Now I keep tossing the coin 500 times.
No,
and
I
it keeps
understand.
coming up
Yeah.
heads, right? Then that seems to me, now there's a more sophisticated variant of it. And there are these combining functions. We should give a presentation on combining functions. Talk about that. No, but there are very several ways to combine, to allow for dependence, I think, right?
You can solve it with machine learning. You don't compute the covariance anymore.
right. Okay, so if you do construct the polygenic score in the standard way that is being done in genetic epidemiology at the moment, and you try to predict education attainment out of sample with the results of our meta-analyzed GWAS, then what you currently get is an R-square of about 2%. Now, interestingly, so the R-square actually gets higher if you include more and more SNPs in your polygenic score, which points towards a highly polygenic trait. What's also interesting is that the score that has been constructed for education attainment actually does a little bit better predicting cognitive function in the same sample, right? So it's 2.5% in R-square. And then we did a couple of theoretical projections for the accuracy of such a simple polygenic score for education attainment, depending on the sample size that you have in your GWAS. And so it looks like that, if you would have 500,000 people to run your GWAS, that the R-square would be 12%, with 1 million would be 15%. An upper bound, we estimated that to be in the range of 22%. So, these are relatively substantial numbers. And, at the moment, of course, it's not possible to run a GWAS on 500,000 or 1 million people. But most certainly it will be possible to do that a couple of decades from now.
Is that for the discovery sample?
Yes, that's.
And then what about the replication sample size? How does that figure in?
They can be very small once you get up to a high R-squared.
Yeah.
So
What's the intuition of why that makes sense, that your replication sample doesn't really matter? I mean, that's what we observed, right? But why does that? Why do you?
Well, so for the accuracy of the polygenic score, what really matters is how accurately you estimate the beta. So the smaller the standard error of the beta, the better is going to be your polygenic score. And so that really depends on the n of your discovery sample. But it doesn't depend on the n of the replication sample.
Just to give a sense of magnitude, if you estimate the polygenic score on 500, if you estimate it on 500,000 people, so you have an R-squared of 12%, you can then get 80% power to replicate it in a sample of like 70 people. So you could do laboratory experiments, using this as a variable.
No, but this makes perfect sense, of course, because theoretically, the hard part is coming up with the best predictor.
Yeah.
It's not.
Yeah.
I mean, once you've got one, now you're describing laboratory amounts of variance. But if I get an effect size of 15% on an experimental test, I can do 15, right?
If someone gives you a black box as a decent predictor, you can tell very easily whether the black box is good or not.
No, right.
Okay. So the big question for us was: is this really is this a real effect, or are we just picking up very subtle population stratification in this study, right? So and that's an obvious problem for basically all GWAS research, but it's probably even more an even more serious problem for something like education attainment, where first of all it's so biologically distal and the effect sizes are so ridiculously small, and it's a trait that is obviously influenced by the environment. So we actually did quite a lot of extra work to get to the bottom of that, and one of the things we did is we tried to replicate our genome-wide significant hits in another very large sample, 23andMe, with a sample size of 34,000. That sample, it was possible to control for population stratification much more rigorously than it was possible in the original GWAS. So instead of just using 4 principal components, what we did in the original study, here we used 25. And what you see are the effect sizes that we found in the discovery stage, which is the dark column. Then in the middle you see the replication phase, and the last column is for the replication sample, so for 23andMe. And obviously the effect sizes are very similar, and we replicated these findings. Then another question was, can we replicate the polygenic scores in even more sophisticated models? So one of them would be a mixed linear model that allows the correlation between the individual's education attainment to be linearly increasing in their genetic relatedness. And we did that in 2 samples, QMR and STR. And basically the R-square that we found in the p-values, they were the same as when we ran these tests in the original paper with more simple methods. And then finally, we also tried to replicate this within family analysis. So we could do that in 3 different samples. 2 of them were in the original paper. And then we did another study with the framing and heart study. And also there, we saw that the polygenic score replicates, although the effect sizes are much smaller than in the other analysis. So it looks like that it's extremely unlikely that the association results that we find are only driven by population stratification. Now, Dan was already hinting at the possibility of using a noisy phenotype as a proxy for something that is underlying or something that is more precisely measured. And one of the things that we try to do is sort of leverage the results on education attainment and trying to figure out if we can use these results to learn something about an endophenotype that makes a lot of sense, which would be a cognitive performance or IQ. And the reason we expected that this could work is because the 2 phenotypes are actually both phenotypically and genetically correlated, and because for education attainment we just had a so much larger sample than for cognitive performance. So what we did is we took our GWAS results, and we basically took out the cohorts from the meta-analysis that had good measures of cognitive performance, and we reran the meta-analysis and came up with a set of SNPs that passed the p-value threshold of 10 to the power of -5. We pruned them for linkage disequilibrium, and then we ended up with 69 independent SNPs. And then we forwarded them to a replication stage, or a second stage, where we had another 24,000 individuals from different samples that had high accuracy measures of cognitive performance. And interestingly, these samples were mostly children that were still, they haven't completed their education yet. So this cognitive performance sample is actually larger than the largest GWAS sample that was available until now. But even that sample size would have been too small to do a GWAS directly. So here's the result as a QQ plot. so clearly there's evidence for association, much more than you would expect by chance. And there are 3 SNPs that survive the conservative Bonferroni correction. Okay, so wrapping up, I think what we've seen until now is that it's very likely that social scientific outcomes that we're interested in are polygenic traits, right? So that they're not influenced by just 1 or 2 genes with a strong effect, but rather hundreds, if not thousands of genes that all have a very tiny effect. Which means that in principle you can discover them, but you will need very large sample sizes. And we're now at a point where it becomes possible to do that type of work, at least for a couple of phenotypes. And this is what we're trying to do now in our consortium. So there are a couple of other projects that we're working on, another stage of education attainment, which will double the discovery sample size. And we also have large-scale GWAS projects on subjective well-being and fertility. Okay, thanks.
Let me first thank all 3.